All right, hey guys, Mr. Johnson here. I'm going to take you through the chapter 6 and 7 notes on Mendel and Punnett squares. This is part 2. Remember, part 1 of chapter 6 was on meiosis, and we compared mitosis and meiosis together. And I think we left off there. I don't really remember clearly the last thing we talked about while we were at school. So I'm going to take you through this note outline. You can follow along with me. You may need to pause me because I'm going to run through it really quickly. Okay. So we're talking about Mendel and we're talking about heredity. Mendel is a person. He's this guy right here. And we'll talk about his little story. So genetics is something we've been talking about for the past couple of weeks. Genetics is just a study of biological inheritance patterns and variation in organisms. And if we know what variation is, variation are just differences in organisms. So how organisms come to be different from other organisms. How you have freckles and somebody sitting next to you doesn't have freckles, or how you have blue eyes when your parents both have brown eyes, or something like that, right? Genetics is a study of those things. Traits are just inherited characteristics. So things like freckles, things like eye color, hair color, types of wings, types of leaves, colors of leaves, or maybe more flowers would be a better example. Those are all examples of traits. So there's some great ones. So plant height is one we'll talk about a lot. And this is a pea plant, and we'll talk about why we talk about pea plants in like right now. So Gregor Mendel, we consider him the father of genetics. He experimentally bred thousands of pea plants together, and he was a monk, so he didn't really have much else to do. He's living in a monastery, and he was a pretty good scientist, though. He designed tons and tons of experiments and then looked at their offsprings. What did he do exactly? Where he crossed? Where is that? Blah, blah, blah. He crossed, or he mated two pea plants together to look at their offspring. He called the parent generation P1, the offspring the F1, and the offspring of that offspring the F2, and so on and so forth. And Mendel observed when he crossed two purebred, which means these are exclusively purple, these are exclusively white. When he crossed those things, he only ended up with purple flowers. Now, why would that be? Well, if we cross purple and white, we'd expect half purple and half white, but what he ended up with was all purple. Then he crossed these, which, yeah, that's kind of weird when you think about it. That's like brother and sister here. He crossed these guys and made them mate, and found that when he made them mate, their offspring was 3 purple and 1 white. So still primarily purple and then 1 white. These were all purple. These ended up being 75% purple and 25% white. So we call it a 3-to-1 ratio. So Mendel observed that when he crossed the F1 generation, the F2 generation produced a ratio of 3-to-1 in most of these traits. And the reason that happens is because these are really simply inherited traits. So Mendel concluded 2 things. The first thing he concluded is that traits are inherited as units. Okay? And the second thing he concluded was the law of segregation. He states that organisms inherit 1 gene from each parent. So 1 gene coming from each parent. Those are the 2 things that Mendel concluded. So what exactly is a gene? A gene is just a piece of DNA that provides instructions, and any alternate to a gene is called an allele. So genes have different options, essentially. So here our options are wrinkled, and the opposite of wrinkled here is going to be round. So wrinkled is an allele, round is an allele. We have both the wrinkled alleles here. Okay, so that means the trait that will be expressed will be wrinkled. Okay. Alleles can be dominant or they can be recessive. So back in this example back here, let's do it. Which one's the dominant? Well, we can just kind of stand back and look and tell that purple is the dominant trait because there's more offspring that's purple, and white will be the recessive trait, right? Okay, so alleles can be dominant or they can be recessive. Dominant is always expressed when it's present. Recessive is only expressed when both the recessive, is only expressed when both the recessive traits are present. Okay? So we'll look into more what that looks like. Alleles can also be homozygous. Homo means same, right? Homologous means same. Homozygous means same. So these alleles are the same, or they can be heterozygous. Oops. So here are some alleles that are the same. We're going to talk about these letters here in a second. But if both these letters are capital, that's homozygous. Also, if both these letters are lowercase, that's homozygous. It's the same thing, right? Two of the same thing. It can also be heterozygous. Heterozygous means different. The only option for heterozygous is a capital letter and a lowercase letter. Okay, heterozygous means different or opposite. All right. So I'm going to pause real quick and I'll come back with you under number 10. So we have different traits here. The first trait is the dominant trait in this table. So tall is dominant, axial is dominant, purple is dominant. We use a letter of the dominant trait when we're setting up Punnett squares. So here, for tall versus short, we're going to use the letter T. We can put that there. Homozygous dominant. So dominant means we have the capital T. Homozygous means the same. So the two letters here that are going to represent homozygous dominant are a capital T and a capital T. This is a homozygous dominant pea plant that is tall. Heterozygous, we only have one option, big T, little t. Heterozygous means different. They must be different. And homozygous recessive, recessive we know is small t. Homozygous tells us the same, so it's going to be two small t's. Now we can do that with each one of these. Axial is dominant, purple is dominant, green is dominant, round is dominant. So we know here are all the letters that we'll use. Okay, you take a minute, pause me, and fill out the rest of this right here. Okay, pause me now. It's going to pop up and it's going to be completed. Here's our chart showing us the genotypes, and we'll talk about what genotypes are. The genotypes of each one of these pea plants. Okay. So what do we do with these letters exactly? Well, we're going to start putting them into Punnett squares. So we're going to go through 11 and 12, and then we're going to go through Punnett and squares, okay? Okay, so all of these letters and all of these alleles and genes make up the genome. So all of your genes make up your genome. It's all the organism's genetic material. So we're just talking specifically about one gene, which is pretty specific, okay? So we have something called genotypes and phenotypes. Genotypes are the actual genes of an organism, and phenotypes are the physical trait that's expressed. So with genotypes, we were talking about just a minute ago in the chart. So for tall versus short, we use those T's. Those are our genotypes. We saw that some can be homozygous recessive, some can be homozygous dominant, and some can be heterozygous. Those are all genotypes. The phenotype is the actual trait. So if it's big T, two large T's, two capital T's, the phenotype is going to be tall. If it's little t, two lowercase t's, it's going to be short. And if it's heterozygous, what's it going to be? Well, it's going to be tall because tall is dominant. So when both tall and short are present, the dominant trait wins out, which is going to be tall. Okay. We're going to skip this, number 13, you can cross off, and we're going to go on to Punnett squares really quickly. So Punnett squares are just a grid system that help us to predict the possible genotypes in a cross. So this is how we predict the offspring. So I left you some space in number 15 to do some practice with Punnett squares. Here's a good practice. We have a homozygous dominant parent, and which one is this? This is homozygous recessive. We set those up in a Punnett square. Parent 1 goes up top. Big T, parent 2 goes down the side, little t. To see what we put in these boxes, all we have to do is look up and look over. Okay, in this box, this comes down, this goes over. Here, look up, big T, look over, little t, there, right? And if we see there's a pattern, it's all going to be the same in this one, okay? This is all the same. And Mendel predicted this cross, right? Okay. So we can say a lot of things. We can say 100% of them are going to be big T, little t. 100% of them are going to be tall, right? These are all monohybrid crosses. So here's another example. Maybe we can write this one down. We've got a big T, little t and a big T, little t. How do we describe these? Well, we describe them as being heterozygous, okay? What goes in this box? We look up and look over. What goes in this box? We look up and we look over. Now, why did I put the big T first? Well, that's just one of our rules. Okay, we don't put the lowercase letter first. We put the capital letter first. Here, same thing, up and over. And here, same thing, up and over. Okay, what do we have here? We have our first homozygous recessive. This is going to be short, while the rest of these are going to be tall. Okay, so we can say 75%, 3 out of 4, 75% are going to be tall, 1 out of 4, 25% are going to be short. That's a 3-to-1 ratio. Here we have another one. You pause me and you do this one. Okay, hopefully this is what you found. And we can use test crosses. I'm not going to go into that. This is a dihybrid cross. Dihybrid crosses just show 2 different traits. This is not as confusing as it looks, but I cannot possibly explain it over YouTube right now, so I'm going to pray that we meet again in the future and that I can explain to you this in more detail. Okay. Oh, probably, I'm sorry, it's a likelihood of something happening. And that, I hope you know what the word probability means. If you don't, here it is. You can write that down. All right, the last thing really quickly, I'm going to go through this very quickly. We have different types of traits, okay? Not all of them are simple monohybrid like we've been talking about, and not all of them fit perfectly into a Punnett square, okay? Specific people can be carriers, which means they don't have the trait, but they can pass that trait on to other people, okay? So for example, a tall pea plant might be heterozygous, which means it has the allele for short, but it doesn't express short. We can talk about that in terms of cystic fibrosis here, okay? Both these parents don't have cystic fibrosis, but they carry the trait for cystic fibrosis, which means they can have an offspring with cystic fibrosis, right? Which this explains how this can happen. Punnett squares predict that pretty good. And this is not a real Punnett square here. This is not how you inherit eye color, but it also really simply explains it, how parents with brown eyes can end up with a child with blue eyes. It's a little bit more complicated than this, but this is a really simplified example of how this can happen, okay? So sex-linked traits, really quickly, are just traits found on the sex chromosome. An example of something that is sex-linked is hemophilia. There's your example, and I'm not going to go through any of this. Okay, this is also something, if you'd like, you can look at this really briefly and try and explain it to yourself, but I don't have the time to do this, okay? We get an incomplete dominance. Incomplete dominance is just a mix between 2 phenotypes. So here we have green-blue. We have a green fish and a blue fish, and we end up with a green-blue fish. Okay, like a mix between green and blue, right? This is like a steel blue, I guess you could say. So we think about incomplete dominance as a mix. A good example, some flowers express themselves in incomplete dominance. And this is a beta fish. Beta fish express their color in incomplete dominance. And then we have codominance. Codominance is when traits express themselves both at the same time, okay? They're both expressed at the same time. So blood type, which is probably something you'll start hearing about in the news really recently with all this virus stuff going on. Codominance is, or blood type is expressed through codominance. So we can have A, we can have B, we can have AB, and we can have O, okay? So blood type is a really good example, and here's that if you'd like to look at that in more detail. Okay, here's another one if you'd like to look at that in more detail. And then a lot of traits are what we call polygenic. Poly means more than one, and that means they're found on more than one gene, okay? Now I'm going to stop there. I'm not going to go into pedigrees. I don't think that's worth it right now. If you end up taking biology in college, which I hope some of you do, I love this stuff, you will get some good instruction on what pedigrees are, and they're not confusing. Essentially all they are is a family tree made up of genes, okay, genes and phenotypes. Okay, hope you guys are doing well. I'm sorry that went on for 14 minutes, but that is all your notes for chapter 6 and 7. All right, wash your hands. Love you guys. Bye.